Hello, anatomists and physiologists. Michelle Glass here, and welcome back to our last cell membrane video, but unfortunately not our last chapter 3 video. What we are ready to talk about here is active transport. And anytime you see the term active and we're talking about the body, you should think about, it's going to require ATP, which remember stands for adenosine triphosphate, and that is our energy molecule. Now, active transport requires ATP because it's going to go against the concentration gradient, and I am abbreviated concentration with CONC period. Because we are going against the concentration gradient, the cell basically gets to determine the direction of movement. It's no longer, we can only go downhill. Now we can go on flat land. Now we can go up the hill, right? So this is what's important about active transport, is that the cell gets to dictate the movement, the directional movement of the material. And then we'll see that active transport is going to require protein carriers. So remember, these are going to be integrated proteins in that cell membrane. And here again is the cell's control, because the cell is making that membrane. The cell changes that membrane up all the time. And so it can determine, what it's going to move by active transport based on those protein carriers. When we talk about active transport, we said already that there's two major types. So we're going to talk about what we can think of as pumps, and then we're going to talk about what we can describe as vesicular transport. And again, the thing that they have in common is that they're active, and that means, of course, that they require ATP. Okay, here again is our memory device, that the significance of active transport is that we're going uphill and we're using ATP, and we can flow against the gradient. We can go from a low concentration to a high concentration, right? Okay, when we talk about pumps, the major one that we should be paying attention to is the sodium potassium exchange pump. Now, this guy is so significant. This pump will be on this exam, obviously. It will be on your departmental final. It makes an appearance multiple times in AMP2. So it's definitely like gonna become very familiar. It's going to become like an old friend. Now, your sodium potassium exchange pump is going to be common on many types of cells. And we're going to see it like really abundant with nerve cells, because we're going to see that it sets up what we can call an electrical gradient. Remember, our term gradient is talking about the grade, the steepness. So what we're seeing here is we're going to set up a difference in charge. Electrical is talking about ions. It's talking about change of charge, movement of charge, right? When we talk about neurons, I'm going to draw like my cartoon drawing here of a neuron. And this would be the cell body. So I have like this really kind of big squiggly line with a nucleus in the middle, and then I'm going to make like this really long extension, which we'll see is called our axon. And here's my whole thing is my neuron here. And when we talk about this electrical gradient, what we'll see in our neuron is that it gives us a slight negative charge inside the neuron of -70 millivolts. So the significance of the sodium potassium exchange pump is that it sets up this electrical gradient, especially in our excitable cells like neurons, and it sets up a resting charge of -70 millivolts. And when we talk about a cell at rest, that means it's not it's not dead, so it's doing stuff. It's doing stuff just like you do stuff when you're sleeping. Your body does stuff when you're sleeping, right? The cell is doing stuff, it's doing activity, but it's not in the process of being stimulated or stimulating another cell, is basically what we're saying. And so a neuron has a resting electrical gradient, a slight negative charge, and we'll see that measure is going to be a negative 70 millivolts. So we'll see that in just a few minutes. The significance of the sodium potassium exchange pump is that it's creating this electrical gradient, is creating this slight negative charge. And it's doing this because we're going to draw like our protein channel. I'm going to draw my pump. I'm just going to create it to look sort of like a cylinder. Okay. And what we're going to see is that it's going to actively pump 3 sodium ions out of the cell, and it's going to actively pump 2 potassium ions into the cell. So it's not equal. We get all kinds of positive sodium on the outside of the cell as a result of this pump. So I'm loading up my extracellular fluid with sodium. I'm loading up my extracellular fluid with sodium. And that's leaving very little sodium inside of the cell. If you survey the body, the majority of the sodium will be in the extracellular fluid. And then I'm going to put some potassium on the inside of my cell. But remember, I've not done an equal exchange. So remember, you have to memorize this. Sodium potassium pump exchanges 3 sodium ions out and 2 potassium ions in. And what this does is collect a whole hell of a lot of sodium on the outside and you know, not quite as much potassium on the inside. And so then remember, opposite charges attract. So anything with a negative charge, chloride ion, proteins with negative charge, they're going to hang out like right inside that membrane because they're super attracted to all this positive sodium on the outside. And so when you measure across the membrane, you get that negative 70 millivolt charge. Okay, this is really the significance of the sodium potassium exchange pump because because the cell has actively on purpose stuck all that sodium in the extracellular fluid. Notice it's creating a super steep hill. It's like when you're in the sandbox and you push all the sand to one side so you have a hill. So now like trucks get to roll down the sand, right? That's what the cell's doing, right? That's what the sodium potassium exchange pump is doing, is creating this sandy hill of sodium, right? So that later it can use it when we talk about neurons firing. So we'll see that again in the future, okay? Now, cells do this active transport right here. Remember, is the pump. And I didn't write it here, but let's, oops. All right, I don't know if you heard it, but my computer is multitasking and a video of osmosis started to play in the middle of this one. So anyway, what I was trying to do was to make sure we know that this process requires ATP, right? Because it's active, because it's a pump, right? So it's using ATP here at that pump in order to do this movement, in order to push all that sand into that hill, in order to push all that sodium out of the cell, you're going to have to use some energy there, right? So let's make sure that we've got that down. Now, what we see here is what we can describe as secondary active transport. And so what this means is it's like indirectly using ATP. So this is like passive transport with an asterisk, right? You have to read the fine lines. So what we see with secondary active transport, in the case of a symporter, you can move two substances same direction. So for instance, here you can move sodium and glucose into the cell, right? And so what we see here is that sodium potassium exchange pump can make that sandy hill, can push all that sodium into the outside of the cell, right? And then it creates that hill, allowing the sodium to move back into the cell using a different channel, and along with it, the glucose, right? So here you can think of like the sodium can sort of like pull the glucose into the cell. Because glucose is often at a high concentration in the ICF. And so that makes it harder for glucose to do passive transport, right? And so sodium is usually low, right? Because of the sodium potassium exchange pump. Right? And so this low sodium is going to help to pull this glucose into the cell. So this is an example of a symporter. So in order for the glucose to come in, you got to take the sodium, and the sodium is easy to pull in because you've created a hill of the sodium on the outside. That's that idea. So it's still like passive, but it's dependent on the activity of the sodium potassium exchange pump. So that's like the fine print that you have to read. Okay, antiporters is going to transport two substances, but now we're going to go opposite direction. So an example here is sodium ion and hydrogen ion. And what we can see here is that the sodium is going to move into the ICF, or into the cell, and the hydrogen will move into the ECF, or out of the cell. And so again, because you've like stockpiled your sodium on the outside, you have an easy time moving it in, which helps to kind of push that hydrogen out. So again, it's kind of like, it's a little bit of cheating, because all of this secondary active transport is indirectly using the ATP because it's dependent on the activity of that sodium potassium exchange pump. So this adds another layer of the significance of that sodium potassium exchange pump. Okay. The next type of transport that we need to look at is still active, but quite a bit different. This is called vesicular transport, and it's using a vesicle. So that's hard to read. Let me make it black. You can think of a vesicle as like a box. It's basically transport, but it's not made of cardboard, of course. It's made of cell membranes. It's basically like phospholipid bundle. And the significance is, you know, just like a box, it's packaged up. So the inside of the vesicle is different from the outside of the vesicle, right? And so this is why it's so important. Now, vesicular transport comes in two varieties. So we have what's called exocytosis, and then we have endocytosis. Now the CYT part of the word cyte means cell. So exocytosis is exit the cell. And endocytosis is basically into the cell, right? And so in the case of exocytosis, we'll see a cell might get rid of digestive enzymes, hormones, the signaling chemical histamine are all examples of things that are going to be exported using exocytosis. With endocytosis, we're going into the cell, and here we have both phagocytosis, which is like cell eating, and then we have penocytosis, which is often described as cell drinking. And what does the cell drink? Extracellular fluid, basically. So in the case of phagocytosis, we see this very often with like our red blood cells. So our white blood cells are able to identify pathogens and engulf or eat them. So a cell that does phagocytosis is called a phagocyte. Most of our white blood cells are a version of a phagocyte, basically. Penocytosis, again, cell drinking, is basically taking in some of that extracellular fluid. And here what we see is sometimes we see this happening where it's not selective, meaning it's just basically like slurping up everything in the ECF. So this is a great way of getting fluid, electrolytes, whatever is there in that ECF. But we also see that sometimes there's like a selective drinking. And so in this case, you basically use a receptor. So what we see is you have receptors on that cell membrane that bind to certain molecules, and then that's what gets brought into the cell. So just to give myself a little bit more room, when we talk about selective or receptor-mediated penocytosis, we see this with the important metal iron that we need for oxygen transport. So we see that it's going to bind to a certain receptor. The iron binds to a molecule called transferrin. And then this is the complex that the receptor binds to from the red blood cell. So the red blood cell will see this complex of not just like loose iron, but iron as paired with transferrin, and that's what's brought in. So it's very specific, very selective. When we look at our vesicular transport, we can use this as an example of exocytosis. So here the cell has, in this picture, I have my cell membrane and I have just a vesicle inside. So that's just a circle with a little bit of like yellow dots. And what we're going to see is that vesicle is going to move until it connects to that cell membrane. And when it does so, it opens up such that the yellow dots, the contents of that vesicle, get emptied into the extracellular fluid. Okay, so we can try that again. So that vesicle packaged with that yellow molecule actually physically moves to the cell membrane, joins up with the outer cell membrane, and then opens up, and that's releasing the contents into the extracellular fluid. The opposite of this is happening in the case of phagocytosis, which would be like cell eating, remember? So here that cell membrane is going to kind of pinch around, and here just for fun, it's pinching around a chocolate ice cream cone because, I mean, why not? I'm sure my cells really like chocolate ice cream. And so basically it's forming this vesicle around that ice cream and then bringing that ice cream inside the vesicle into the cell. So this is important if instead of ice cream, we were actually drawing, like E. coli or some pathogen. You wouldn't want that pathogen to be able to infect your cell. You'd want it to be packaged up. And then remember, penocytosis is like the cell drinking. And so again, just for fun, and excuse me, instead of engulfing an ice cream cone, we're engulfing a cup of coffee or hot tea, and it's that same process. Okay, that's it for cell membranes. Stay tuned for organelles. And as always, take care of yourselves and each other.